The last speaker of this session is Professor and Senior Consultant Jens Petter Berg from University of Oslo and Oslo University Hospital. You have an MD from 1986 and a PhD from 1995 and research interest which is quite broad but focused mostly on endocrine diseases. Please.
So, good afternoon everybody, and thanks to the organizers for inviting me to give this brief introduction to metabolomics. And yes, omics is more than genomics. It includes also metabolomics, and I will give you some examples. My background is that I am head of the Department of Medical Biochemistry at Oslo University Hospital. I am, among other things, responsible for some of the ancient tests from the 50s that the former speaker referred to. They are still quite good, I must say, and we use them. But we will also take into use some other new tests, and I will share some of them with you. So, what is metabolomics? Well, you can say that it is the collection of all small molecular compounds in a biological sample. We typically restrict it to a molecular weight or mass of less than 2,000 Daltons. And among these compounds you can find that you will use for studies of the metabolome or plasma. It may be urine, it can be a cell, it can be in a tissue or in a whole organism. And you have also different kind of metabolomes that you can study. For example, the studies of all lipids, which is generally called the lipidome. You can study the glycome, which is the study of carbohydrates, for example, and sugars. We use studies of metabolites regularly in our lab, but then generally in a targeted way, that we study one particular metabolite, such as glucose, creatinine, cholesterol. These are typical metabolites. When we perform metabolomics, we do systematic studies of the metabolites and metabolite profiles. And this term is often used synonymously with metabolomics. And the approach can be targeted or untargeted. That is, we may have an opinion of which compounds that we are mostly interested in, or we can have an hypothesis-free approach and use an untargeted method. That is, we don't know exactly which metabolite to look for, but the methods that we use may help us in identify new targets. And the two basic methods that are used are mass spectrometry, which we've heard of previously, used in, for example, proteomics, but also we can use nuclear magnetic resonance spectroscopy. This is how typical biochemical pathways wall charts may look like, and you've probably seen it. They hang around in the laboratories, and they are probably from the 60s or 70s or something around that. That is the 1960s and 70s. And it is also quite fascinating to follow, for example, the route of acetate turning into, for example, cholesterol. But no wonder really that during the, for example, 90s and 2000s, that students chose rather to go into molecular biology, where you only need to know 4 different bases compared to this maze of different compounds and reactions. But nonetheless, at least from a metabolomic centric point of view, we can regard the omics cascade in the following fashion, where we start with a genome, which can tell us about what can happen. And then we have the transcriptome, which tells us what appears to be happening. And then we have the proteome, which we heard of, and the proteins are what makes it happen. And we end up with the metabolome. And as we like to say, it tells us what has happened and is really happening. And together these make the phenotype. And even when you combine these different omics, you can call it the interactome. And the lability of the metabolome is important to have in mind, especially when you plan a study. by holding your breath for just a minute or so, your metabolome will change dramatically, whereas the genome will be quite stable and in a new fashion. I should have maybe changed the picture a bit. Maybe the whole world is holding the breath and not this guy right now. But enough of that. From a clinical chemist point of view, we divide an analytical process in different phases. And we often talk about the pre-analytical phase, in which all factors concerning biological variation must be taken into account. And then we have the pre-analytical phase, which concerns sample collection, transport, handling, and storage, such as Frode referred to. We have the analytical phase, where test and method quality is concerned, and also the specificity and sensitivity of the methods that we employ. And then we have the post-analytical phase, in which we have to interpret the results in a good way. Now, the pre-analytical phase, there we have some factors that we can do, that we can control, and some we can't control. For example, age and gender and gut microflora are difficult to control, whereas dietary effects, we may have some control of which kind of diet the study persons are having. We can control the sample collection time when we know that the metabolome is changing diurnally. That is, we can see differences in the metabolomic profile if a sample is taken early in the morning compared with late in the night. And also physical activity is possible. It is possible to have some control on what kind of physical activity the person has had the few hours before the sample is taken, at least. Now, the pre-analytical considerations, then we know that the concentrations of several of the metabolites can change during sample handling. And this may be due to that there still exists some enzymatic activity in the sample. And this enzymatic activity may degrade or transform some of the substrates that are small metabolites, and you may produce some other substrates during just handling of the sample. For example, in urine samples, there may be bacterial activities that can also change the metabolomic profile of the sample. Then we have chemical reactions that may not be enzymatically driven, but can be, for example, accelerated by exposure to air or by exposure to light. So we have to have some control on these, and in order to be better to handle such samples, there is a development of so-called evidence-based standard operating procedures, and the Da Vinci European Biobank initiative has some advice in a metabolomics-driven infrastructure, and you can visit their website and get some information on how to best preserve your samples for metabolomic studies. And these, I have a pointer here. This is a so-called standard operating procedure. It's recommended for handling urine samples, and from collection to centrifugation step, you should have the samples at no more than 4 degrees, and there should be no more than 2 hours from collection to centrifugation. And this is important to remove cells and particles, and also the handling here to reduce the effects of any possible enzymatic cellular or cellular activities on the metabolite profile in the sample. And then for storage, it is recommended for urine that it is on liquid nitrogen or liquid nitrogen vapor. And this is to avoid breaking any residual cells that may not have been removed by the centrifugation or an filtration step. Of course, this is a very rigorous operating procedure. So it is important, if you have any deviations from a procedure, always to make note of it so that you know how the sample has been handled. For plasma or serum, it is recommended that either EDTA or citrate are used as anticoagulants when you want plasma. Otherwise, you may collect serum, and then you have to clot the sample, and it should be clotted for at least half an hour or at least 30 minutes before you can do the centrifugation. And also remember to keep the samples cold. And then you process the serum or plasma by centrifugation. And if you're interested, for example, in the lipidome, the lipids are subject to oxidation, and then you have to have an inert atmosphere in order to preserve the lipids. Especially phospholipids are subject to degradation and oxidation. Samples should be frozen immediately after processing, and long-term storage should be performed at -80. And so to the analytical phase. And you have done the sample preparation, and you are ready to analyze samples. And then you have basically two different options. It is the mass spectrometry with either gas chromatography or liquid chromatography, which are can be quite tedious when it comes to sample workup and preparation. Whereas NMR spectroscopy is quite simple when it comes to how to prepare the sample before analysis. When you analyze the samples, you will buy NMR spectroscopy and get these peaks as depicted here, whereas gas chromatography or liquid chromatography will give these mass over charge map. Post-analytically, you have to have some procedures in order to identify metabolites. You process the data. You can do some unsupervised or supervised statistical analysis. For example, principal component analysis, and also loading plots is used in NMR spectroscopy in order to identify metabolites that are of particular interest. And you can identify potential biomarkers, and of course you often have to do some validation experiments afterwards when you have your candidates. I'll just give you some examples from studies that have been performed in collaboration with Frode Riise and Daniel Sakse at University of Oslo. We have used the NMR spectroscopy, and the spectroscopy is usually really a large magnet, and here we have the samples that are fed into the magnet. And we measure the resonance of protons, that is hydrogen atoms, in the compounds. When we use this for imaging, we are interested in, or the radiologists are interested in, the water signals. We are not interested in water signals. We are interested in the signals from the other compounds that have hydrogen atoms bound to them. And this is a typical spectrum that we can get of protons from a urine sample. Here we have spikes that are from format. Here we find hyperate, for example. Here we have urea, glucose, creatinine, alanine, for example. So we get this spectra. And in a study where we used NMR spectroscopy, was this in collaboration with Nakstad. She's interested in different resuscitation protocols that are used on newborns with respiratory arrest. So she set up this study in which 125 newborn pigs were subjected to asphyxia until they had a cardiac arrest. And then they started 6 different resuscitation protocols and also took different blood and urine samples from the pigs, which we studied for magnetic resonance spectroscopy, metabolomics. We had samples from before the asphyxia was started, 2 hours after return of spontaneous circulation again, after a cardiopulmonary resuscitation had been performed on the pigs, and also 4 hours after that again. And by using just the information from the spectra without any knowledge of which kind of compounds are hidden in the spectra, we can set up these metabolic trajectories in a principal component analysis. Here are individual pigs denoted by these different numbers, and you can see that the pigs generally lay in this area before the asphyxiation. Then 2 hours after return of spontaneous circulation, they are in this area, in the green spots, and then 4 hours after, they return to this bluish area, but not completely back to the starting point. There were no differences between the different resuscitation procedures, and then we could also not identify any differences in the metabolic profiles of the pigs. But we can go into more detail and look at which metabolites were changing during these procedures. And this is from the starting point and to 2 hours after return of spontaneous circulation. And we do this kind of loading plots where we actually subtract the we have the profiles obtained at this point 2 and subtract the profiles obtained at point 1. And then we find that there is an increase in lactate signals, there is an increase in alanine signals, and also in glycine, pyruvate, glutamine, alanine here, and lactate again, and these are branch chain amino acids. Of course, there is no big surprise that lactate increases when you asphyxiate someone. And you can also explain this by going into the biochemical literature, that you have that is asphyxiation and increase in lactate and alanine can be explained by activation of these basic cycles, the Cori cycle in which lactate is put out from the muscle because of glycolysis and non-aerobic degradation of glucose. Lactate is transported back to the liver where it is forming glucose again. So this is the lactate increase, and we have the alanine increase by this glucose-alanine cycle in which muscle produces pyruvate, which is converted to alanine, which is exported to the blood and in the liver taken care of, and then reconstituting glucose, and which is exported and sent to the muscle again. So no big surprises there, really. In another study we performed in collaboration with Anne Karen Gjenum, we studied the occurrence of gestational diabetes or diabetes in pregnancy in a cohort of a little more than 800 pregnant women in the Stork-Groruddalen study. So we had urine samples from visit 1, which is at week 8 to 20 in pregnancy, at visit 2 at week 28 in pregnancy, and also a sample at visit 3, which is at 12 weeks postpartum after delivery. And the test for gestational diabetes was performed at week 28 with an oral glucose tolerance test and measurement of fasting plasma glucose levels. And this is also the metabolic trajectories of these pregnant women and also after delivery. We can see that at visit 1, the cluster in this area, and then at visit 2, we have moved a little bit up and to the right. And after delivery, we move to the left and a little bit more up on the scale. And we can, by a certain amount of security, differentiate between samples taken, for example, at visit 2 and visit 3 with 99% certainty based on the metabolic profile alone in urine. But we were not able to identify gestational diabetes by this principal component analysis and multivariate analysis. However, we could decompose by univariate analysis the differences between visit 1, 2, and 3 by just 2 metabolites. An unknown signal, which we found, which is depicted along the x-axis. It is probably related to placenta function. And along the y-axis, the excretion of lactose, the main sugar in breast milk, the excretion of lactose in urine. During pregnancy, these women develop mammary glands and milk production, and there is also some leakage of lactose into the circulation, and also which is also then excreted in urine. And we can see that it increases from visit 1 to visit 2, and also increases further to visit 3. After delivery. And we also see that at visit 3, some of these women have a rather low excretion of lactose in urine, and these are the women who are not breastfeeding their infants. By using univariate analysis, we are also able to find that the excretion of citrate in urine was associated with a more pronounced form of gestational diabetes mellitus. We see here from visit 1 to visit 2 that there is an increase in citrate excretion among women with a more pronounced hypoglycemia. And this normalized after delivery, which is also what we see with the gestational diabetes. The women have no diabetes after delivery, but they have an increased risk of developing type 2 diabetes later in life when they have had an episode of gestational diabetes. And when it comes to breastfeeding, we could also look further into some other analytes or metabolites there. For example, these are the level of breastfeeding, entirely, partly or not, along the x-axis at week 12 postpartum. And then we measured looked at a particular metabolite at week 28 in urine and found that the level of glycine excretion in urine in women who were later not breastfeeding after delivery was lower, had a lower excretion of glycine in their urine compared with those who were entirely breastfeeding or partly breastfeeding. Why glycine should play a role in whether a woman would breastfeed her child after delivery, we are not quite sure. But this is hypothesis generating, and glycine is an important molecule in the one-carbon metabolism. It fuels the so-called one-carbon metabolism and goes into this folate cycle, which is important, among other things, for producing nucleotides. So maybe it has something with cell division to do, we don't know. But the really interesting things, I think, come when we combine the metabolomic information with, for example, genomic information. And this has been done by several authors, and this is a rather big study by Sure and co-workers, published in Nature some years ago, where he published this map of human metabolic individuality, where, for example, you could see an association between the levels of the amino acid glycine, for example, in plasma, with the gene CPS1 and genetic variants of this gene. And so it could, they found several metabolites which could be linked with genetic variances in specific genes. And this interactome can be done to the extreme, and it has been published in this paper in Cell, in which the last author, Michael Snyder, performed his personal omics profiling. It is called, he called it IPOP, an individual personal omics profile, an IPOP. And at several visits, he had a whole genome sequencing. He had polymorph of his white blood cells. He had a whole transcriptome sequencing. He had a proteome profiling. From serum, he had an untargeted proteome profiling, a targeted protein profiling using a cytokine array. He had a metabolome profiling, an autoantibody profiling, and also several medical and lab tests. Altogether, he analyzed in himself 2.7 billion RNA fragments, more than 6,000 proteins at 14 time points, and 6,800 metabolites, and revealed by this that he had an increased risk of diabetes, although he had no traditional risk factors for diabetes. And this was revealed in his second virus infection during this study, that he had an increase in his glucose levels and also an increase in his hemoglobin A1C level, which is a marker of long-term glucose exposure to hemoglobin. It is a rather expensive way of finding out whether you have a liability to develop diabetes, I must say. But maybe it could be of some interest, at least as a principle. Someone also called it that he had developed a narcissome by doing this personal study. But I mean, it could be interesting. So, to summarize and conclude, metabolomics, systematic studies of all small molecular compounds in a biological sample cell tissue organism. And we have basically two methods that you can use, either mass spectrometry or NMR spectroscopy. Mass spectrometry is rather a targeted way of doing it, whereas NMR spectroscopy is basically untargeted and hypothesis-free. The advantage of mass spectrometry is that it is more sensitive. It goes into the nanomolar or picomolar range, whereas NMR spectroscopy works in the millimolar to micromolar range when it comes to metabolite concentrations. It is important, I will stress it again, that sample collection and handling procedures have to be adjusted to the purpose. I mean, if you want to study a lipid, the lipidome, you have to make sure that there is no oxidation of lipids when you handle the sample. And also, I would say that meaningful analysis afterwards requires a relatively large number of samples because you get a large number of metabolites to analyze. We have, unfortunately, no core facility for metabolomics in the Oslo area, but there are core facilities in Trondheim and also in Bergen. And by that, I will conclude and hope that you will find your favorite metabolite or pair of metabolites or profiles that you can work with, and wish you all the best of luck in your projects looking for new biomarkers among metabolites. Thank you for your attention.
Questions, comments? Jessica?
Well, thank you very much. I don't know whether you were present yesterday or not.
No, unfortunately not.
Because Mats Melby, he is also at Stanford, where Snyder is, and he presented this method, but that was on pregnant women, sampling every week, doing the full transcriptome, and it is a very fascinating approach. And as you say, it's expensive, but as a hypothesis generating method, I think it's quite fascinating. Since biobanks are entirely a compromise, I mean, at least if we want to build big biobanks with huge sample collections, we have to compromise. There could be 250 kilometers from where the sample is drawn and where it's frozen. So it's what we in Norwegian would call the muliges kunst.
Yes, absolutely.
Yes. Yeah. So I was assumed that most of these studies where metabolomics and genomics have been combined, if you go back in the literature, are based on biobanks where this has been handled in different ways. And actually, UK Biobank, they did several pilot studies up front when they started their sample collections to figure out what was the best way of doing metabolomics using serum or plasma or urine, and what was the effect of leaving the sample at +4 for 12, 24, 36 hours before the analysis was done. And it was not impossible. There are still a number of opportunities. So actually, I would suggest that since we have now had some great presentations about different methods, and your slide showing that metabolomics is actually a functional follow-up of all the genotypes that we have present. We should meet as soon as possible and develop large-scale protocols for how we can utilize this as soon as possible, and then test also the ability to retrieve all the data from the different registries. So let's do that.
I completely agree, and we should not be. We should be aware of the limitations, but not be hindered by them. We should be. We should know that there are limitations, and we should. But the project should take care of it and focus on the areas where we have less limitations, at least. And I think that would be very useful when you plan new biobanks, that all omics expertise from all omics areas are included in planning, so that we know where the limitations are, at least.
Just a comment from me. What I hear is something I quite like, that the last talks interact very heavily with genomics, and that you're starting to talk and getting things together, because it is connected. Another reflection is that the metabolic scheme was quite a bit simpler when I went to medical